Okay, good morning. We'll just get going, and then when the coffee gets here, we will officially take a break. Oh, yay. Maybe let's take a break first.
We're going to leave it out here.
Okay. But does anyone besides me need a cup of coffee? Okay. Let's get a cup of coffee, and then we'll start. Much better. Okay, so now we'll officially get started. So as Nicole said, I'm here today from CSU, where I'm sort of Nicole's counterpart at Colorado State University. So I run the core facility there. And what I'm going to talk to you about today is, again, as she said, it's going to be a little bit redundant of probably what you saw in your online training. But basically, the goal of the next 2 hours is to give you the basic overview of a proteomics workflow. So what are the key points of your sample preparation steps, what happens in the mass spectrometer, and then how do we deal with the data analysis. So very bare bones, very quickly, but give you an overview, and then we'll go into each part in more detail in the labs and over the next couple days. Okay? So first of all, we can ask the question, what is a mass spectrometer, and what does a mass spectrometer do? And in very basic terms, a mass spectrometer measures the mass of ionized or charged molecules, right? Very basic definition of what a mass spectrometer does. And why is this useful to us in proteomics? In proteomics, this can be useful for example, for determining the masses of peptides or intact proteins. And this can be used through what we call peptide mass mapping to identify proteins. Now, the ability to measure masses can also be used for more specific applications. For example, the determination or localization of post-translational modifications. Because if you're modifying a peptide sequence, you're changing the mass of that amino acid. And so that change in mass is something we can detect with a mass spectrometer. Okay? Now, there have been a few key technological advances that have really allowed this field to grow into what it is today. And the first of these is in the area of mass spectrometry itself, and specifically in the development of soft ionization techniques. So this is electrospray and MALDI, which I'll talk about later in more detail. And the big deal here is that these soft ionization techniques allow you to ionize a molecule without fragmenting it. Okay, so that's what allows us to measure the intact mass of our fragile biological molecules like peptides and proteins. So in addition to these ionization techniques, there's been dramatic increases in the sensitivity and mass accuracy of mass spectrometers. And this just keeps getting better and better. Every year, they just get better and better. And maybe more important than that is that mass spectrometers have become much more user-friendly. So it used to be, even I would say 5 to 10 years ago, that pretty much the only people using mass spectrometers were the people who they went to grad school and they got their PhD, like they were experts in mass spectrometry. And now any one of you, as you're going to do this afternoon, could go into a lab and operate a mass spectrometer and get data out because the vendors have made them much more user-friendly, which has opened them up to a much more diverse group of researchers. Okay, so the next big advance has been in the availability of protein sequence databases. And the vast majority of information in these databases comes from translated genomic sequences. So really, the large-scale genome sequencing projects had to come first before we could do large-scale proteomics experiments. Okay? And then along those lines, the third big advance has been the bioinformatics tools. And by this, I specifically mean the database search engines. Because it's one thing, we have these great mass spectrometers, we can generate all this data, and we have these enormous protein sequence databases, but how do you merge the two? How do you take this mass spectrometry data and actually mine those protein sequence databases to get anything useful out of it? And that's where the database search engines come into play. And we're going to talk about database searching towards the end of my lecture today. But one thing I should mention here is that this is still a very developing field. So we can, at this point in time, generate much, much more data than we can actually analyze. And you'll see this throughout the course of your experiments in the lab as well. Okay, so here, do I have a pointer? Yep. Here's our basic proteomics workflow that I think you've already seen. We're always going to do some level of sample preparation, and there are some key steps here, even though this is going to vary greatly depending on your application. There are some key steps that are going to be common to every proteomics workflow, and we'll talk about those in a little more detail. Then you're going to acquire your data. So this is your mass spectrometry analysis. Then you're going to take your data and you're going to do some sort of database search and get a protein ID or other specific information. And there can be a lot of additional bioinformatics analysis that goes into this after this point. But this is your basic workflow. OK, so we'll start with sample prep. OK, so as I said, there are a lot of steps that can go into sample preparation. And again, this depends on what the goal of your experiment and the type of sample you're working with. You're always going to do some level of simplification, OK? And this could be done in many different ways. One way to simplify your sample is to do gel electrophoresis. Another way is to do liquid chromatography fractionation. And we'll talk about many specific examples of sample fractionation in the lab this afternoon or this morning. But the key here is that we need to simplify the sample, and I'll talk a little bit more about why that's important in a little bit. The other key step in your sample preparation is protein digestion. And so we're going to talk about that in a little bit more detail later. OK, so we're going to talk about that in more detail right now. So digest your proteins. What does that mean? It means we're taking our protein and we're using an enzyme to break it into smaller pieces that are called peptides. And the peptides are what we're actually going to measure in our mass spectrometer. So why is it necessary for us to do this? There's a few reasons that this is necessary. The first of which is that most mass spectrometers have limited mass ranges. And this is a very general statement because all mass spectrometers are different. But most of the mass spectrometers that are widely available for us to use have a limited mass range in which they can measure masses accurately. So that's part of it, okay? But probably a bigger part of it is that you can imagine that even if you could measure the mass of your intact protein really, really accurately, within a couple parts per million accuracy, there still could be quite a few other proteins that have that same intact mass. So it's very hard to confidently say, when I definitely have this protein, that's what this is, based on one measurement, even if it's a really good measurement, right? However, if you take that protein and you digest it into fragments, the pattern of peptides that you get from an enzymatic digestion is going to be sequence specific. So this fingerprint of peptides is going to be a more confident way to identify your proteins, because you're going to have 5 to 10 measurements that are specific to the sequence of that protein versus one intact protein mass measurement. Does that make sense? Okay. So those are the reasons that we digest. The other step that I didn't mention earlier that's unique, or that's common to all sample preparation workflows, is cleaning up your sample. Okay? So many of the things that you might do to your sample up front, you lysing your cells, doing whatever fractionation scheme you choose, are going to introduce substances into your sample that aren't mass spec friendly. Things like salts and detergents in particular. And so we always do some level of cleanup to get rid of these contaminations before they go into a mass spec, or it can really tank your whole analysis and also really screw up the mass spec. So, and then the other key step in your sample preparation workflow, again, is to simplify your mixture, okay? And the reason for that is, the main reason for this is that as great as mass spectrometers are, they can still only measure a given amount of molecules in a finite amount of molecules in a given amount of time, right? So if you use the example, let's say that your mass spectrometer is only capable of analyzing 10 molecules at a time. And that's not true, but for simplicity, right? So you have 100 molecules in your sample. And if you take that whole sample, inject it in the mass spectrometer all at once, you're only going to see 1/10 of what's there. But if you were to take those 100 molecules, and before they went into the mass spec, you fractionated them into 10 fractions, and then each of those fractions were analyzed separately, then you, in theory, could see all of the molecules in your sample, right? So if you imagine a whole cell lysate, and you have thousands of proteins, and each of those proteins is then digested into 10 or 20 or 30 peptides, I mean, you have an enormous amount of molecules in there. And so you are going to be, if you inject all of that into your mass spec, you're going to be seeing a very small fraction of that. And the other key part to that is that in addition to only being able to detect a certain number of molecules at a given time, there's also dynamic range limitations. So in a whole cell lysate, the range of protein concentrations that are going to be there are going to span many orders of magnitude. A mass spectrometer is generally limited to about 3 to 4 orders of magnitude at the most that it can detect at a time. Okay, so by spreading out those proteins, you're going to have a better chance of seeing some of the proteins that are sort of at the lower concentration levels. You're going to expand your dynamic range. Okay, so now we're going to talk more detail about protein digestion. In our workflows, in proteomics workflows, you're almost always going to use trypsin to digest your proteins. Trypsin is a digestive enzyme that works by hydrolyzing peptide bonds, and the reason that we like trypsin so much is because of its specificity. So trypsin is very good at cleaving pretty much only at the C-terminus of lysine and arginine amino acid residues. Okay? So what this means is that if we have a protein sequence like this, and we were to digest this protein with trypsin, we can predict the peptides that are going to form. Because every time there's a lysine or an arginine, trypsin will cleave this peptide, or cleave the protein. And if we can predict the peptides that are going to form, we can predict the masses that we should observe in our mass spectrometry analysis. Okay? So this is a very key, this ability to predict our enzymatic products is a very key part of our proteomics workflow. And we'll talk more about how this factors into our data analysis at the end. How do we make this digestion most efficient? Okay, so in order to have an efficient digestion, your cleavage sites, so those are your lysines and arginines, must be accessible to the trypsin. Okay, and so in order to do this, we have to fully denature our proteins before the digestion. And we can do that either by running them in an SDS page gel, which is a very good way of denaturing your proteins. Or if you're in a solution situation, you can use a denaturant such as urea or guanidine. Okay? Now, in addition to unfolding the protein, we also have to be conscious of disulfide bonds. Okay? So if you have multiple cysteines in your protein sequence, they can form disulfide bonds that will sometimes keep the protein in a semi-folded state even under denaturing conditions. And so what we do is we add a reducing agent such as DTT and an alkylating agent such as iodoacetamide. So this will reduce the cysteine bonds and then cap them with an alkyl group so that they won't reform in the process of the digestion. And then after these two steps, we would add our enzyme. Okay, so in more detail, this reduction and alkylation step. So here's our protein. It's already been denatured, but it's got a couple disulfide bonds, so it's still partially folded, and trypsin is going to have a hard time getting into some of these sites in these folds to cleave this protein if we didn't denature it. So we add DTT, we open this protein up, and now we've got these reduced cysteines. We add our alkylating agent, and now we have our modified cysteine residues. Now, the reason I show this in detail is because this is an important consideration in our workflow. By doing this reaction, we are chemically modifying all of the cysteines in our protein, which means that we are changing the mass of all the cysteines in our protein. And this particular reaction changes the mass of cysteines by 57 Daltons. And so when we are analyzing our data downstream, we have to remember that we did this and take this into account. Otherwise, we're going to miss every single peptide that has cysteine in it if we're not taking into account that its mass has changed. Yes, sometimes we do use different enzymes. Now, I would say 90% of the time we use trypsin, because if you're trying to do protein identification, almost always trypsin will work. It will give you enough peptides to identify the protein. And it's the most specific, and so it will give you the best database search results. Now, there are some situations where you need to increase your coverage, your amino acid sequence coverage of the protein. For example, if you're targeting a specific post-translational modification, and when you digest that protein with trypsin, the amino acid that has that modification is in this tiny little 3 amino acid peptide, we're never going to see it. So you will have to modify your protocol in order to get a good digestion. For example, in my lab, we do a lot of histone work. And so we're doing analysis on histone acetylation, which is on lysines. And so trypsin is not very good at cleaving at lysine if it's modified, but it will sometimes cleave. So it confuses the analysis. And so we routinely use RHC for histone proteins when we're specifically looking for lysine acetylation. Okay? All right, so few pointers for protein digestion. And I believe you're going to be doing a digestion this afternoon in the lab, so Rick will go over this with you in more detail. One of the biggest things you can do to tank your whole experiment is contaminate your sample with keratin. And if you've worked with a core facility at your institution, you've probably heard this already. But keratin is the protein that's in your skin and your hair. It's everywhere. It's in dust. It's all over labs. And so a little tiny bit of that in your sample will just overwhelm your protein and generally ruin your analysis. So you have to be very conscious of keratin contamination. And so in our lab, what we do is we have a hood that's set up just for protein digestion. And it's not a laminar flow hood or a cell culture hood. It's a regular hood, but it's a sort of contained space that we can keep clean. And so before we start digestions, we go in with a methanol Kim wipe and we dust, basically. You wipe everything down. You wipe the pipettes down. You always wear gloves. If you have long hair like me, I'm always shedding hairs, and I try and pull my hair back. But you have to just be really conscious of it. And also, if you're running a gel as a precursor step, be conscious of that too. When you're destaining, don't cover it with saran wraps so things don't settle on your gel. So if you take precautions, it's easy to avoid, but we get samples all the time that are just keratin. And it's just disappointing because you have to do it again. So again, dedicated lab equipment. And so if you have buffers that you use for digestion, just have those just for digestion, because a lot of times someone else is going to use them, they're going to leave the cap off, and then dust is going to settle in there, and So that's all I'm going to say about sample prep. You'll get a lot more in the lab this afternoon. Are there any questions before I go on? Okay. So now we're going to talk about the meat of our workflow, our mass spec analysis. Okay. So as I mentioned earlier, there are a lot of types of mass spectrometers out there. And you'll learn about many different ones, and you'll get to use a few different mass specs in the lab as a part of this course. But in general terms, all mass spectrometers have 4 parts. They have an inlet, which is how you get the molecules in. They have an ionization source, and this is where we ionize the molecules, because a molecule has to be ionized in order for us to detect it. Then it has a mass analyzer, and this is where we separate the ionized molecules over time so we can detect them one at a time. And then we have a detector. So we'll talk about each parts of the mass spectrometer separately. So the inlet, again, is our method of sample introduction. Now, there are 2 major types of inlets that you can use. The first is a solid state inlet, which is either some sort of plate or a probe. The other is going to be a liquid inlet, which is going to be an infusion or an injection. And then the choice, which one you use, is dependent on, what's available to you, the type of mass spectrometer that's downstream. And by mass spectrometer, we usually define the mass spectrometer by the mass analyzer. So when I say type of mass spectrometer, I'm talking about the type of mass analyzer downstream. And also the sample complexity will have a great influence on which type of inlet you use. And I'll talk about that in a minute. So plates or probes, these are, again, solid phase inlets. These are used primarily with single component samples or very, very simple mixtures. Okay? Because in this situation, each of these spots on here is a separate sample, and everything on that spot is going to go into the mass spectrometer at one time. There's no more fractionation up front. there's no way to couple any type of fractionation with this because it's solid. So it can't be very complex because everything's going to go into the mass spec at one time. Infusions or injections, on the other hand, are liquid phase, and so they are almost always coupled, especially if it's an injection, with high-performance liquid chromatography. Okay? So in this case, before the ions or the molecules go into the mass spectrometer, we can hook up a liquid chromatographer to the mass spec inlet. And so we can separate the molecules over time, and then they'll go into this inlet, liquid inlet, one at a time. Well, not really one at a time, but a few at a time, as opposed to with a solid state where everything goes in at once. Does that make sense? Okay, so we can utilize a liquid inlet injection for more complex samples because we can couple it with liquid chromatography. Okay. So what is the experience level here with liquid chromatography? How many people have used it?
You say liquid, like just a column with beads in it?
Well, sure.
Okay, I've done that.
Okay, so I'm going to go ahead and then go through this review of HPLC. So again, why is it important to separate our molecules? Because we can only look at a few things at a given time, and so if we have a very complex sample, we need to separate it before it goes into the mass spec. So chromatography gives us an additional level of separation of the peptides before they go into the mass spectrometer. What we are almost always using when we talk about, so when you hear the term LC-MS or LC-MS-MS, what they're meaning is liquid chromatography coupled to your mass spectrometer. That's what I'm talking about here. And in that situation, we're almost always using what's called reverse phase liquid chromatography. And the HP stands for high performance or high pressure. So this is where it's different than your column that you're doing manually. In this type of situation, we're using pumps to push our solvent through a column. So it's higher pressure as opposed to using gravity, right? Okay, so that's what HPLC is. So high performance liquid chromatography. So in reverse phase chromatography, what we're doing is we're separating our peptides based on their hydrophobic nature. Okay? And in this scenario, the peptides and proteins are going to bind the column initially in an aqueous solvent, and then they will elute off over time as we increase the organic solvent that's flowing through our column. Okay? So we use our HPLC pump, so this is where the pump comes in to actually create a gradient of increasing organic solvent under pressure. Okay, so here for example, over time we increase the amount of acetonitrile that we flow over the column. So the columns that we use in this type of chromatography are packed with these silica particles that are derivatized with a stationary phase. And in reverse phase, this is going to be some sort of hydrophobic molecule. And this can be generally C4, C8, or C18s. For peptides, we're always using C18s. If you're trying to do intact protein reverse phase chromatography, you don't want something that hydrophobic because then it's really hard to get the proteins off. So for proteins, we might use C4 or C8. And we'll talk about that, I think, more in the lab this morning. So what you've got are these silica beads with these little hydrophobic tails sticking out all over them, and then your column is packed with all of these. Okay, so here's our little column, and it's packed with all these little particles with these sticky C18 chains on them. We have loaded our peptides onto the column here. So if we load them in an aqueous buffer, they're pretty much all going to interact with this C18 material, and they're going to sort of stack up at the front of the column. And then over time, as we start to increase this a percentage of organic solvent, basically what happens is you get displacement, right? So the peptides that are are not very hydrophobic are going to, it's not going to take much organic solvent to displace them from the column. And so those are going to elute first. And then the really, really sticky peptides, you're going to need a lot of organic solvent to sort of take up all those binding sites and kick it off and elute those. So those are going to come out last. So over time, you separate your peptides, and as they come off of the column, they go straight into your mass spectrometer. Okay, so I'm going to move on to ionization sources. So we've got the molecules in there. Now we have to give them a charge. Okay, so again, we're doing soft ionization here, which means that we're going to get no significant fragmentation in the ionization process. Okay, and we're almost always, while you can do you can create negative ions and look for those, we almost always look for positive ions, which is from protonation. And that's because we will want to fragment these later on in the mass spectrometer, and I'll talk about that. But the positive ions fragment better in the mass spectrometer. So, and it's generally a more efficient process to create positive ions than negative ions. Okay, so there are two types of soft ionization. There's MALDI, which stands for Matrix-Assisted Laser Desorption Ionization. And there's electrospray, which stands for electrospray ionization. Or sorry, ESI, which stands for electrospray ionization. So I'll talk about MALDI first. Now, MALDI is a solid state ionization. So this is going to be coupled with our probe or plate inlet. Okay? And in the MALDI process, what we do is we take our sample of mixture of peptides, and which are these lines, and we mix it with a matrix, which is a UV absorbing compound. We mix this together, and then we spot it on our plate or probe and allow it to co-crystallize. Okay? Now, then what happens is we irradiate the sample with a UV laser. The matrix ions absorb the UV laser. Everything sort of explodes off of the plate. And then you get a proton transfer in the gas phase from the excited matrix ions to your sample molecules. And so what you end up with are gas phase, singly charged sample molecules, as well as matrix molecules. And these will all go into the mass spec at one time. So in this situation, there is no liquid chromatography. So everything on your plate, it gets ionized at once and all goes into the mass spec. So this works well when you have very purified samples. So for example, you did a two-dimensional gel and you have your protein, or a one-dimensional gel and you have your protein isolated to a single band or a single spot. You will likely couple this with MALDI analysis. Now the advantage of this, and I don't know if I go into that, no. The advantage of this is that it happens really, really fast. It's pretty much instantaneous. So you don't have that 40-minute gradient to get your mass spectrum. You hit it with a laser, boom, you have a mass spectrum. So it's very, very fast. Okay, so electrospray ionization is our other type of soft ionization. And in this case, again, we're doing liquid phase. So most likely we've got an HPLC out here. And then our liquid that contains our peptides is flowing through this little tip. And these are really small. Nicole will show you this. They have a little video so where you can see the little tips. But they're really, really small. They're like hairs. And it's pulled fused silica, actually. And so the tip is very fine, generally about maybe 30 microns in diameter. And what happens here is that you apply a voltage to this tip so that as the fluid comes out of this charged tip, you get this plume of charged solvated sample molecules. Okay? So this plume of droplets then goes through a region of drying gas, and the solvent starts to evaporate. And as the solvent starts to evaporate, the charges, the positive charges on the surface of this droplet get closer and closer and closer until they repel each other, and then the droplet explodes into smaller droplets, and then this process keeps happening until all the solvent is gone. And what you end up with in this situation are multiply charged gas phase molecules. Okay, so because of how this ionization process works, you rarely see singly charged ions. Almost always 2 or 3 or sometimes 4 for peptides. And for proteins, you can have 20, 30, 40 charges on a protein. And you basically end up with a Gaussian spectrum of peaks that represents that protein in all the different charge states. Okay, so when would you use one versus the other? Again, some of it's dependent on what's available to you. And really the biggest factor in this choice is your sample complex or is it not, right? If it's not complex, if you've got a single protein band or it's 2D gel spot or a purified, lipid or something like that, we can do MALDI very, very fast. And that's going to be probably your best bet. If you have any sort of mixture, you're going to want to do electrospray because of the ability to couple with HPLC. Okay? All right, let's move on to the mass analyzer. Okay? So again, this is the part of the mass spec that separates the ionized molecules over time. And this is the part of the mass spec that sort of defines what the mass spec is. So what does a mass spectrometer do? A mass analyzer do? A mass analyzer separates or resolves our ionized molecules based on their mass to charge ratio, which is called M/Z. Okay? So what this means is that if we have a molecule that had so our peptide has a mass of 1,000 Daltons, and this was done by MALDI, so we have a charge state of 1. The mass over charge, or the M/Z, that the mass spec is actually going to detect is going to be this 1,000 Daltons plus 1. So the charge comes in the form of a proton or a hydrogen, which adds a Dalton to your mass. So you have 1,000 plus 1 divided by 1. So the M/Z of this peptide is actually 1,001. That's what we'll detect. Now, if we were doing this by electrospray, so now we have a charge state of 2, the same peptide is going to look like 501 to us because we're going to have 1,000 plus 2, because now we have 2 protons, divided by 2, because we have 2 charges, to give us 501. So we will actually see 501 in our spectrum, but that corresponds to a peptide of 1,000 Daltons. Okay, so there are lots of types of mass analyzers. Some examples here: quadrupole, time of flight, which is called a TOF, an ion trap, FTMS, a new type of mass analyzer, which is called an orbitrap. See, I do it too. I call them mass specs because you name them based on their mass analyzers. These are what are called single mass analyzer instruments. You can also have hybrid mass analyzer instruments, which are basically 2 mass analyzers in tandem. So this would be an example, for example, the quadrupole time of flight, or a QTOF. So you'll use a QTOF in the lab today. A tandem time of flight, or a TOF-TOF, or a quadrupole ion trap, which is called a Q-trap. So we're going to start by talking about time of flight. And then I'm going to also talk about quadrupoles, ion traps, and quadrupole time of flights. And these are the types of instruments that you're going to get hands-on experience with in the course of this class. Okay, so I like to start with time of flight because I think it's the easiest to understand. Based on your very basic undergraduate physics class or chemistry class, everybody knows this equation: kinetic energy equals 1/2 mv squared, right? So in our time of flight mass analyzer, what's happening is that we are going to generate our ions here in our MALDI source, and you can have an electrospray source here as well. And just in this example, I used a MALDI source. So we generate our ions, and then we're literally measuring the time that it takes for the ions to fly from the point of ionization down what we call a flight tube to a detector. Okay? And the way we correlate this time measurement with mass is based on this equation. So we're trying to determine mass, so we can solve this equation using our basic algebra for mass. And then we look at our variables here. So v is velocity, and this corresponds to, as you know, meters per second. In this equation, the meters is going to be the length of our flight tube. Okay, and this flight tube is under very high vacuum. So this all occurs under vacuum. And we know the length of this flight tube very, very accurately. Our seconds is our time of flight. So that's the time it takes for the molecule to fly down the length of the flight tube. And then this is the mass of our ion. Oh, and then the one I apparently didn't point to was the kinetic energy. So kinetic energy is actually a constant. And what happens is that at the point of ionization, we give all of the ions a voltage boost so that at time 0, all of the molecules have the same kinetic energy. Okay, so this is a constant. And the length of the flight tube is a constant. And what we're measuring is the time of flight. And so we can use this measurement to calculate the mass. Okay, so based on this relationship, the mass is inversely proportional to the velocity. So the small ions are going to travel the fastest, and the larger ions are going to travel more slowly. Okay, we're going to hit it with the laser. Oh, and there it goes. And see, the little one, it went really fast, and the big one went really slow. I worked hard on that. Okay, any questions on this? Okay.
That's awesome.
Except that is awesome. That's right. So this is what a spectrum looks like that you get out of a time-of-flight mass spectrometer. Your x-axis in your mass spectrum is always, the units are always m over z, or mass over charge. Okay? So, and then your y-axis is always intensity. And these units here are very arbitrary. And this is one thing I'm not going to go into in this lecture, but I think later Nicole gives you a lecture on quantitative analysis by mass spectrometry. And what I'll say is that mass spectrometry is not a quantitative technique. And the reason for that is that the height of this peak is based on how many ions we detect. But how many ions we detect is based on both how much is there and also the ionization efficiency of that particular molecule. How well does it take on a charge? Okay, so in this example, this was one protein that was digested with trypsin, was analyzed by MALDI. So in theory, we should have equal amounts of all the peptides from this one protein. But we obviously don't. And that's because some of them ionize really well, like this one. Some of them ionize really poorly, like these down here. And some of them don't ionize at all. So it's not quantitative, and Nicole's going to go into that more about how you can deal with that and still use it for quantitative analysis. Okay, any questions? All right. So the next type of mass analyzer that I'm going to talk about is called a quadrupole mass analyzer. This was probably one of the earlier mass analyzers to be developed. And this type of mass analyzer acts as a mass filter. Okay? So it literally are 4 rods. That's where the quadrupole comes in. 4 poles. And we apply electric field on these rods such that only ions of a selected mass over charge ratio are going to have a stable oscillation down the length of the quadrupole. And so those will be the only ones that will make it the whole length and come out the other end to the detector. The rest of them will become unstable and go off on the side. Okay? So it's very good as a mass filter. Okay, so we rarely use quadrupoles by themselves because they're really good mass filters, but for reasons I'll go into later, they're not that useful for proteomics, mostly because they're good at mass filtering, but they're a little bit slow, and also the mass accuracy, which is an important part of our process, is not very good. And also, you can't fragment the ions. And so that's what I'm going to talk about now, is that we don't fragment the peptides when we ionize them because we want to be able to see the whole peptide. But we do want to have the ability, okay, once it's in the mass spec and it's ionized, now in a controlled way, I want to break it apart and look at how it fragments. And so this is where we might use one of these hybrid mass analyzers. So we have 2 mass analyzers in tandem. And so a very common configuration in proteomics is what's called a quadrupole time of flight. So we're taking these 2 types of mass analyzers and we're sticking them together. The quadrupole is the first one, and this acts as our filter, mass filter. So we can select a specific peptide. Okay, so we say, okay, I only want to look at this one peptide. I'm going to send it into this collision cell where I'll add a gas like hydrogen or nitrogen, and helium or nitrogen, and the addition of that gas will cause this peptide to fragment. Okay? And then all of the fragment ions will travel through the TOF, where we can detect them based on their mass to charge ratio, and we can detect the masses of all the fragment ions. So this is an example of what we'd call an MS-MS, or a fragmentation spectra. So in this case, if you remember, I had a starred peptide. I didn't point it out in our last, when I showed you the mass spectrum. So in that spectrum, all of the peaks were different peptides. In this spectrum, what we've done is we've told that quadrupole in our front end to isolate one peptide out of everything that's there. So we set the filter to say, there's all these molecules here, but I only want to look at this one. So filter for this peptide, allow it to go into that collision cell. Then I'm going to break it apart by adding my collision gas. And then in the TOF, what we analyze then is we still sometimes have some of that initial peptide left. And then all of these peaks are actually smaller fragments of this particular peptide. Okay? Now, this is very, very important to our workflow, and I'll talk about this more when we get to the data analysis. But this is important because this fragmentation process is very predictable. Okay? And again, this is nice because while we have different peptides, they're all peptides. They all have a peptide backbone. And this mechanism of fragmentation is really dependent on the peptide backbone. And so the fragments that we get are going to be specific fingerprints for that particular peptide sequence. So it's the same idea that we can predict the tryptic peptides. Now we have a fingerprint because we know where they should fragment, right, when we digest with trypsin. So now we have a fingerprint of peptides for that protein. It's the same with our fragmentation in the mass spec. We have a peptide, we know where it should fragment, and now we experimentally have a fragmentation pattern, so we can predict and we can match them up. The next type of mass analyzer I'm going to talk about is the ion trap. Okay, so we've talked about the time of flight, the quadrupole, and then the quadrupole and the time of flight put together to give us our hybrid instrument. Ion traps are unique in the sense that they are a single mass analyzer, but they can do both MS. They act like a hybrid. They can do MS and they can do the MS-MS fragmentation all in one. Okay? So the way these work are very similar to a quadrupole, except for the fact that instead of letting ions of a particular mass to charge cycle through, you can trap them. So it's like a quadrupole with two caps with a cap on each end. So you put the molecules in there, and then you put a cap on them, and then you can get rid of the ones you want, but the ones you don't want, but the one you want stays in the trap. Okay, so we have another little animation. So this is another cute animation, which I can't take credit for. Rick did this one. So we have our protein, we digest it, we ionize it, and then it goes into the trap. Okay, so right now what happens is we put all these ions into the trap. We've trapped all of them. Now we start changing the electric field on these quadrupoles so that they get ejected one at a time. Okay, and then as they get ejected, we detect them, and then we create our mass spec. So this instrument has been calibrated so you know that at this electric field, it will eject this mass. Okay, so then what happens, the next step of what we can do is we can fill up the trap again. So now we had all those peptides in there, and then we said, okay, eject them, but don't detect them. Just get them out of there and isolate the one we're interested in. So now we've isolated this one ion in our trap. We add a collision gas, we break it apart, and then we say, now let's cycle through those electric fields again, slowly so that we can detect the ions one at a time, and we have now created our MS-MS. Okay? Okay, so this is an example of a spectrum from an ion trap. So here was the MS scan. Okay, so all these peaks are different peptides in our sample. We isolated this small peptide in our trap, and we fragmented it, and then this was the resulting MS-MS fragmentation pattern. So then, the next time it might refill the trap and isolate this one, and then collect an MS-MS, and then isolate this one and collect an MS-MS. So this is happening so fast, And you can imagine over the course of even a short 30-minute gradient, so you've got your HPLC, you're separating your peptides over time, and they're going into the mass spec, and you're continuously doing this over 30 minutes. So you create thousands and thousands of spectra, even in a relatively short run. Okay, so the last mass analyzer that I have here is what's called an Orbitrap. And this is in the same class as what are known as FTICR mass specs, or FTMSs. And these are higher end, very high accuracy mass specs. So FTMS instruments are like NMRs. They have big giant magnets, which is what makes them really, really expensive. And I don't think I have an FTMS slide. Nope. This schematic here is of an Orbitrap mass spec. And Orbitraps are very similar to ion traps. This is a relatively new type of technology. This was only released or developed about maybe 4 years ago. So these are relatively new. But basically, it's a type of ion trap in which the molecules are injected into what's called an Orbitrap, which is like a trap, but instead of ejecting the molecules out to measure their mass, they create them in orbit. So they put them in orbit using electric fields. And then they detect the frequency of orbit, basically. And then they Fourier transform that, similarly to how the NMR works, into time. And then they can calculate a mass from that. And so Nicole does not have one of these, so you won't get to use one, but they're becoming more and more common. FTMS instruments, because of the need for the magnet, are very expensive, probably ranging anywhere from 700,000 to a couple of million for these instruments. Orbitraps, what's great about them is that they can get similar levels of resolution, but they are in the range from, like, the most expensive Orbitrap is going to be about 700K. Okay, so they're becoming more and more popular and making this concept of high resolution or high mass accuracy mass spectrometry more generally used. So you'll see it a lot in the literature, that more and more people are doing this, and it's sort of where the field is going as they become more and more affordable for the general researcher. Okay, and that was just supposed to be a little primer about the cool stuff, but that we're not going to use in the class. Okay, so then the detector is some sort of form of electron multiplier. We're just detecting ions and then turning that into a signal, amplifying that and turning it into a signal. There's nothing fancy about it. Okay, oh my gosh, I am so far behind. So I want to talk quickly about some key terms when we're thinking about mass spectra. And the first one is isotopes. So we know that our peptides are composed of chemical elements. All of our elements have naturally occurring isotopes with predictable abundances. And our mass specs can resolve or distinguish between these isotopes. And the isotope we're most concerned about is carbon, or C13. So 1% of all naturally occurring carbon is C13. So C13 differs from C12 by a whole Dalton in mass. Okay, so we are going to see that. We're going to see that shift or that effect. And we're going to see that like this. So these spectra are all single peptides. So a peptide does not just look like one peak. It has an isotopic pattern. Okay? So, and the pattern changes based on the size of the peptide. This peptide right here has only 70 carbons. And so your pattern is going to look like this. The first peak of your pattern is always your C12 peak, which we call the monoisotopic mass. This is the peak where all the carbons in the peptide are C12. And when you only have 70 carbons, it's most probable that all of the carbons are going to be C12. So your pattern is going to look like this. So here's the peptide where one of the carbons is C13. Here we have two C13s. As we start increasing the number of carbons, at some point we get to a level, and this is around 150 carbons or more than 2,000 Daltons in molecular weight, where all of a sudden now it's more probable, it's more likely that at least one of them is going to be, at least one of your carbons will be C13 than none. Okay, so the probability that at least one of them is C13 is greater than the probability of none of them being C13. And so your pattern starts to shift. So now your single C13 peak is the largest, but this is still the mass that we care about. That's still the monoisotopic mass. And then eventually you get to the point where you get above about 3,000 Daltons, where now the second isotopic peak is the biggest. But this is still the one we're measuring. So this factor right here is one of the reasons why our mass range is limited. And it depends on the mass spec you're using. But once you run out of exceed the capacity or the ability of your mass spec to distinguish the resolution between the isotopes, you can no longer measure this monoisotopic mass. Okay? So once this goes into the noise and you can no longer detect it and distinguish it from the others, that's the end of your mass range for peptides. Okay? In terms of measuring accurately the monoisotopic mass. So this introduces the concept of resolution. And resolution is how well you can distinguish one peak from its neighboring peak. And so a high resolution instrument, such as the green or the black trace, is going to be able to distinguish this isotopic pattern and allow you to measure the monoisotopic mass. A low resolution instrument, or when you get past that mass range where you can get this resolution, is going to give you what we call an average mass. So it's going to give you one peak, and you're going to calculate your mass at the apex, which is going to be average of all the isotopic contributions. Okay? So that's the concept of resolution. An extension upon this concept of resolution is the concept of mass accuracy. So the more resolved your peaks are from one another, generally, the more accurately you're going to be able to measure the mass of that monoisotopic peak. And in mass spec world, we talk about mass accuracy in units of parts per million, which is calculated as the error in the measurement divided by the actual mass times the million. Okay? So for example, if we have a 1,000 Dalton peptide and our error is plus or minus 2 Daltons, that would be considered a 2,000 parts per million mass accuracy. And I don't know if there's any mass specs that are that bad. So low resolution is generally going to be right here. And this region right here is going to include mass specs like your ion trap or your single quadrupole. Okay? Those are considered low accuracy instruments. They're going to give you about plus or minus 0.5 Daltons or 500 ppm, somewhere between 100 and 500 ppm. Okay? As we start getting down here to high resolution region, so here in this example, if we have 1,000 Daltons and we measure this plus or minus 0.01, then that's considered 10 parts per million. Okay? This is where you're going to have your time of flight instruments. Somewhere between 2 ppm and 10 ppm is where your time of flight instruments are going to be. Those are considered relatively high resolution instruments. Your Orbitraps and your FTMS instruments are going to be 2 ppm or better or lower. So you're going to see those terms a lot when you're talking about different mass. People will throw those around when they're talking about different mass specs, resolution and mass accuracy. Okay, and this is just showing you the difference in what a spectrum looks like going from lower resolution to higher resolution. And this is from an Orbitrap. So here's, this is 7,000 resolution going all the way up to 100,000 resolution. And you can see how the peaks get sharper and sharper and sharper. Now, but this answers your question here. This is going from, to collect this spectrum, it took 0.2 seconds. To collect this one, it took 1.6. So there's always a trade-off between time and sensitivity. Okay, now again, sorry, PC to Mac there, translation. Sensitivity is a harder thing to quantify, and because the sensitivity of your instrument will depend on the mass spec itself, but it also depends on your sample, right? So when you see sensitivity specs for a certain mass spec, they have taken something that ionizes really well and is very, very pure, and they've seen what's the lowest we can go and still detect it. Okay, but there are a lot of other factors that affect sensitivity, such as the complexity of the mixture and how well your molecule ionizes. Okay, but in general terms, you can think of, so nanomoles is just like child's play. That's easy. Picomoles is routine. So if someone brings me a sample, they say, how much should I give you? I say, if you bring me a picomole, I can see it. For sure. Femtomoles, I say hard, but this is really routine too. So for example, in our lab, we use BSA as a standard protein for QC on our instruments. And when we do our QC BSA injection, we're injecting 200 femtomoles of BSA, and it's a beautiful signal. So this is really, femtomoles is really pretty routine as well nowadays. The slide's kind of old. Attomole is where you're starting to get hard, but still doable depending on the sample. And when you say sensitivity, it also depends on what you're trying to see. We could probably definitely see attomoles of BSA. We just might see 1 or 2 peptides. At 200 femtomoles, we get 60% coverage. Okay, and then zeptomoles, rarely reserved. So you can see that even at femtomoles, maybe we're not that far away from like a single ion. So the sensitivity is quite good of most of these instruments. And then I think there's this, which I've already said. Okay, so which mass specs are you going to use? Again, a lot of it's what's available to you. Here we've tried to make a little table that sort of summarizes these. What's available to you, and also how much money you have, because obviously as you go up in resolution, the cost also goes up, right? And if you need MS or you need MS. So I just tried to summarize the different types of mass specs here for your general reference. Okay, so you guys ready to speed through database searching? So we're going to talk about database searching real quick. And then I think I have a lab with you this afternoon where we'll do database searching so we can go back over some of the things that I speed through. So we've prepared our samples, we've analyzed them in the mass spec, we've created thousands of these spectra. Now what do we do with them? We have to analyze them. So we'll go to our database searching workflow where we're going to take our mass spec data, we're going to submit it to a search engine, and then the search engine is going to query a protein sequence database and give us a result. So we'll first talk quickly about the protein sequence databases. So where does the information in these databases come from? Some of it does come from direct protein sequencing by Edmond degradation. But if any of you guys have ever actually done this, it's quite a tedious process, not really high throughput by any means. So by far, the most of the information in there comes from translated genomic sequences or translated DNA sequence. We do sometimes utilize express sequence tags or EST databases, and these are your single-pass cDNA sequences. They're not full-length sequences. They generally have a lot of errors to them. But if you're working with a species that's in its beginning stages of its genome sequencing, then oftentimes this is all that's available. But you don't want to use them unless you absolutely have to. Okay, so there are a lot of sequence databases out there. I have a few examples. So the largest, all the protein sequences databases that are out there are in the public domain, meaning that they're free. So anyone can go and download them. The largest of these is the one maintained by NCBI, which you've probably been to that website for whatever reasons where PubMed is. And they maintain a very large public database. This is a very old, I think. It's well over 8 million sequences now. And when we do a live search this afternoon, we'll see where it's at today. I think of it as basically a compilation of many public domain databases. It's sort of like a repository. There's nobody monitoring what's getting dumped into this database. So it's not curated at all. So there's a lot of redundancy, duplicate entries, and errors. SwissProt, on the other hand, is a database maintained by the Swiss Proteomics Society, which is a European government-run society. This is a curated or manually filtered database. So the level of accuracy in this is very high. There's virtually no redundancy. But it's obviously a lot smaller. They can't keep up with all the sequences that have been published. But the quality of the data is much higher. And this is also an old number, but I still think they're under 300K right now. Or maybe close. And then we have our EST databases, which are huge and obviously not curated and have a very high error rate and sort of our last resort. Okay, the one thing common to all of these protein sequence databases is that they all use what's called a FASTA format, which is shown an example of a FASTA entry right here. This type of entry, it starts with a caret, and then it contains your accession number. So this particular entry is cross-referenced, as they often are, between different databases. So here's your SwissProt accession number, and then IPI is International Protein Index, and that's another database, so it's cross-referenced. It's going to have a taxonomy ID number, and so this is specifying the species that this protein is from. And some database search engines use that as a filtering device. It's going to have the protein name and the protein sequence. So this is important to know if you want to create a custom database or a custom entry to your database. So oftentimes someone will come to us and they'll say, well, I have this protein, but I mutated it. So we go into the database and we go in here and we change one of the amino acids, or we just create a new entry. All right, so we're going to talk about the search engines now. So we have these databases. They have lots of information. We're going to about to searching. So how does a database searching work? There's two types of searches you can do. The first type is where we're using only the MS data. So these are where we just have peptide masses. And this type of search is called a peptide mass fingerprint. Okay? And in order to do this type of search, which is becoming less and less common, honestly, you have to have analyzed your sample using an instrument capable of high mass accuracy. Okay? So you would never do this search in conjunction with an ion trap analysis, for example. Okay, so you have high accuracy measurements of your peptides in your sample, but you haven't fragmented them. So we're going to identify the protein based on peptide masses. So we have our protein, we've digested it with trypsin, we analyze the peptides, and then what we do is we literally extract into a text file the masses of all the peptides in our spectrum. We submit that list of experimental peptide masses to a search engine. And what does it do? The first thing that it does is a theoretical digest of all the proteins in the database. Okay? So you say, okay, I know I have a human protein. So let's look at all the human proteins. Let's theoretically digest them with trypsin. So we create lists of theoretical tryptic peptides based on cleavage at lysine and arginine for each peptide, each protein in the database. And then we look for the best match between our experimental peptide list and all of these theoretical tryptic peptides from the database. This is done in sort of an iterative fashion. So let's say you've submitted your text file with experimental masses and you start looking and trying to match, and at first you might find up 100 proteins that have this mass. But then you start narrowing it down, and maybe there's 1,000 proteins that had the one mass. Maybe there's only 100 proteins that have both of these masses. And then hopefully, and usually, there's only one protein that matches all of your experimental. So it matches the fingerprint. Okay? Now, more commonly, what we're doing almost exclusively these days is what's called an MSMS search. Okay? So this is where we're searching the fragmentation spectra. So in this case, obviously, you have to have an instrument that's capable of generating fragmentation spectra, which almost all of them are now. So this would be either an ion trap or some sort of hybrid mass spectrometer. This type of search is going to give you sequence-specific information for each peptide, because remember I said that that fragmentation pattern is dependent on the sequence of that peptide. Whereas if we're just searching based on peptide mass, it's still a fingerprint of tryptic peptides, but we're still only measuring a mass. So in this case, the big difference here is that we're actually identifying peptides, not proteins. So we identify peptides, and then those peptides are grouped together to form a protein identification. So here's a schematic that describes how a peptide fragments in a mass spectrometer. So here's our peptide backbone, and this particular peptide has 4 amino acids, right? So R4, 3, 2, 1, okay? In the mass spectrometer, we're going to get preferential cleavage just based on bond energetics and the energetics of the collision right here at this amide bond along the peptide backbone. Okay? If the peptide breaks here, you're going to get two halves. The half of the peptide that contains the C terminus is called a Y ion, and the half containing the N terminus is called the B ion. Okay, so in a population of these peptides, you never just have one, you have a bunch of these same peptides in your mass spec. Some of them are going to fragment here, some will fragment here, some will fragment here, and then when you sum all of the resulting fragments together, what you get is your MS-MS spectrum, or you get basically a ladder of fragment ions that correspond to cleavage here, and then cleavage here, and then cleavage here. So if your resulting spectrum is basically a ladder where you're going to have Y2, Y1, Y2s, Y3s, and so on if it's a longer peptide, and you'll also have corresponding B ions sometimes. And so you can imagine then that basically if you take the mass of this Y2 ion and subtract the mass of this Y1 ion, the only difference between this and this is the residue R3. So the masses of these fragment ions are specific to the amino acid sequence. And these are different ions that are less. You sometimes will get cleavage here, and that's called an A ion. And different types of fragmentation will give you C and Z ions, and it's all based on the energetics of the fragmentation. But in everything, in what's called collision-induced dissociation, which is everything I've talked about today, you're going to get preferential cleavage here. So in a real example, this is a peptide that has 12 amino acids, and it was fragmented. So here's the fragmentation pattern. Here's our intact peptide here, which we'll call Y12. If we cleave the peptide right here, this part contains the C terminus, so it's the Y ion, and that's going to be Y11 because it's got 11 of our amino acids, and it's Y because it has a C terminus, so that's Y11. There is a corresponding B1 ion that's down here that we just didn't detect. Here, if we cleave here, it's going to be your Y10, and that's shown right here. And then you would, again, have the corresponding B2, which is actually right there. And there's your Y9, and so on. And if we look at the mass difference between our Y10 ion and our Y9 ion, it equals 101.1, which is the residue mass of threonine, which is that residue right there. Okay? So the process of doing this, where we're looking at the mass differences between the ions, is called de novo sequencing. This is, in practice, very, very difficult. And this is because even this is a relatively clean spectrum. But you can imagine, at the very least, you're going to have corresponding ladders of Y ions and B ions. So you first have to figure out what's a Y and what's a B. Then, as she pointed out, you sometimes will get a random A ion in there or a random C ion in there, which happens sometimes, but not very often. And then, to complicate it more, you oftentimes get loss of water or loss of ammonia from these ions. And so figuring out what all, because there's a lot of ions in there that we haven't labeled as Y ions or B ions. So it becomes very complex in trying to figure out what's what. And so you sometimes can get strings of maybe 2 or 3 amino acids, but getting the whole sequence is very, very difficult. So in reality, what we do is database searching. So again, we've done our experiment. We've created now, our text file is no longer a text file. It's going to be some complicated XML type or format where we have information now about the parent peptide, what the mass of the parent peptide was, what the masses of all the fragments were, and also something about the charge state. Because if this is done in a electrospray ionization, you're going to have multiple charges. And so you have to know, because one thing you can notice here is that you have fragment ions that are higher M over Z than your parent. And that's because your parent could have 2 charges, so its M over Z is small because it has 2 charges. Fragments are typically singly charged. So you have to know what the charge of your parent was. So what does the search engine do? It does, again, the theoretical tryptic digest of all your proteins. So we have lists of theoretical tryptic peptides. Then it goes one step further, and it theoretically fragments all of those tryptic peptides. So it says, what are the Y and B ions that I could get if I were to fragment this peptide into my mass spec? And then, again, it looks for the best match. And here we're looking, again, we're identifying the peptide. Okay, so we're matching experimental fragment ion spectra to theoretical fragment ion spectra. Then we've identified all these peptides, and we say, okay, this peptide matches to this protein, and this peptide matches to this protein, so I've identified this protein. And that's how we identify the peptide. Now, the basic rule is that if we're just identifying a protein based on the masses of the peptides, we have to match at least 5 of these peptide masses to say we have confidence. It's 10 o'clock. I think I'm going to skip. I have not yet talked about the scoring algorithms. We can talk about all of this afternoon. So everything that I've talked about right now is very general. Okay, so in general, all of the database search algorithms work this way. But there are many, many different software packages out there, and probably all of you are going to have access to different ones. And so, and they all are going to work a little bit differently, mostly in the way that they score the results. So they're looking for matches, and then the search engine has to give you some level of how confident am I in this match. And that's where the search engines differ. So we can talk about that this afternoon, and we can base that conversation on each group, what types of analysis software you might be using or have access to.